Happy morning, children. Today we'll start structure of a mature human sperm. A sperm has 4 parts: head, neck, middle piece and tail. In this diagram you can see from here to here, this is head. This constricted portion is the neck. After neck, this is middle piece and then is the tail. So these are the 4 parts of a sperm. Head contains a cap-like portion at its interior end called acrosome, and posteriorly it has an elongated haploid nucleus. Okay, so this is the part head. Tip of the head is occupied by a cap-like structure called as acrosome, and the remaining part of the head is occupied by an elongated nucleus. So head has only 2 parts, right? Now, one thing I want to tell your children, that previously we studied about spermatogenesis, in which there was formation of spermatid non-motile structures, right? And these non-motile spermatids, then they get converted into motile sperm. So how a non-motile structure, just like an ordinary cell which has a nucleus, which has mitochondria, which has centriole, gets converted into a motile sperm, right? That process is spermiogenesis. So when a non-motile structure like this ultimately gets converted into a motile sperm, right? So what happens actually? The Golgi body occupies the topmost position, forming acrosome. The nucleus gets enlarged and occupies the major portion of the head, right? After that we come to another part of the sperm, that is neck, present in between head and middle piece. This neck is very small and neck has 2 centriole, right? The centriole which is towards the head region, this centriole, this is proximal centriole. This one is proximal centriole, and one which is towards the middle piece is the distal centriole, right? So just like an ordinary cell, a sperm also has plasma membrane, it has nucleus, it has Golgi body, it has centriole, it has mitochondria, right? So this is the neck which contains the 2 centriole. Okay, the centriole which is present towards the head, which is called as a proximal centriole, it has a role in the first division of the zygote, in the first cleavage of the zygote, right? What is the function of proximal centriole? It plays a role in the first mitotic division or first cleavage in the zygote. And the distal centriole, which is present towards the middle piece, it gives rise to the axial filament of the tail. Axial filament of the sperm is produced by the distal centriole, right? Now after this neck piece, there is middle piece. From here to here there is middle piece, and this middle piece actually is occupied by the mitochondria present in the cell, right? And since this region contains mitochondria, so what is its function? Its function is to provide energy for the sperm movement, right? Now one more thing you can see over here, that at the end of the middle piece there is a centriole named as ring centriole. Got it? But this ring centriole does not have any known function. Still, scientists have not been able to find out its function, right? And then comes the last part, that is the tail. From here to here, this is the tail. Tail tail is several times longer than the head, right? And its main function is to help the sperm in motility. It helps the sperm to move in a fluid medium, right? Now, a human male ejaculates 200 to 300 million sperms during a coitus. A human male releases 200 to 300 million sperms during a coitus, during a sexual intercourse. Out of these 200 to 300 million sperms, for normal motility, at least 60% must have normal shape and size. 60% of the sperms should have normal shape and size, and out of these 60%, 40% should show vigorous mobility, so that from the time they are being discharged in the vagina, they could be able to reach up to the site of fertilization, up to the fallopian tube, right? And again we have heard about the term semen. So what is semen? Semen is a collective term used for seminal plasma secretion by all three glands of the male reproductive system, that is seminal vesicle, prostate gland and Cowper's gland. So a collective secretion of all three glands along with the sperm is called as semen, right? So this is spermatogenesis. This is the structure of a sperm. Coming to the oogenesis, that is process of formation of egg or ova, right? What is the site? It takes place in the ovary, right? But there is a difference between spermatogenesis and oogenesis. Spermatogenesis starts with the onset of puberty, but oogenesis starts in the fetal stage. Fetal stage matlab during the embryonic development of the fetal ovary, then only the process of oogenesis starts. Okay, how does it start? Some cells in the germinal epithelium of the ovary of the fetus, हम लोग अभी एम्ब्रियोनिक स्टेज की बात कर रहे हैं, किसी और स्टेज की बात कर नहीं रहे हैं, क्योंकि we have already seen that oogenesis starts in the fetal ovary when the fetus is inside mother's womb, right? एम्ब्रियोनिक डेवलपमेंट में ही ये इसका फॉर्मेशन शुरू हो जाता है, right? Some cells in the germinal epithelium of the ovary of the fetus are larger than others, right? And these cells which are larger than others, they divide by mitosis and produce more of their own type. And what they are called? They are called as egg mother cells or oogonia. Okay, and what is the ploidy of egg mother cell? Egg mother cells are diploid, 2N. Okay, and what will they do? They will only form egg, right? So no more oogonia are added after birth. Oogonia are also called as egg mother cell. They are diploid, right? These oogonia start cell division when in the fetal stage only, they start cell division in the fetal stage only, right? Enter, matlab meiosis 1 starts, they enter into prophase 1 of meiosis 1, but get temporarily arrested. The division in the oogonia stops at prophase 1 of meiosis 1, or now they are called as primary oocyte. Got it? फिर से just listen to it carefully. Oogonia, where are they present? They are present in the germinal epithelium of the ovary. What is their ploidy? They are diploid, they are 2N. right? These oogonia start meiosis 1. Okay, they enter prophase 1, but the division is stopped. And now, at this stage, when this division has stopped, meiosis only has been initiated. They are called as a primary oocyte. And what is the ploidy? It is 2N. Right. One more thing, these primary oocytes get surrounded by a layer of granulosa cells, and they get transformed into primary follicle. Okay, so you see, primary oocyte is a single cell structure like this. What is the ploidy of primary oocyte? It is 2N. This primary oocyte gets surrounded by a layer of cells, and this structure now is called as a primary follicle. ठीक है, तो ये primary oocyte है, and primary oocyte has got a layer of granulosa cells attached to it, and the structure so formed is now called as primary follicle. लेकिन primary follicle has primary oocyte inside it. Okay, and this occurs in the fetal stage only. A large number of primary follicles degenerate during the phase from birth to puberty. Got it? A very large number of follicles, primary follicles, degenerate from the birth to puberty, and this process of degeneration is called as follicular atresia. So that only 60,000 to 80,000 per follicles are left in each ovary by the time of puberty, right? Out of so many primary follicles developed, most of them degenerate, leaving only 60 to 80,000 primary follicles per ovary by the time of puberty, right? Now, each primary follicle gets surrounded by some more layer of granulosa cells, right, and a new theca and a covering, and this structure so formed is called a secondary follicle. Okay? Once again, let us continue with the same diagram. Primary oocyte, which is only a single cell structure where meiosis 1 has stopped at prophase 1. When this primary oocyte gets surrounded by a layer of granulosa cells, it is called as primary follicle. When this primary follicle gets surrounded by some more layer of granulosa cells and a theca covering, the structure is called as secondary follicle. मतलब primary follicle और secondary follicle, what is the difference? Secondary follicle will have some more layers of granulosa cells, and it will have a covering, it will have a theca, and inside secondary follicle you can see what is this? Primary oocyte. Okay, next, a secondary follicle soon transforms, changes into a tertiary follicle characterized by the presence of a fluid filled cavity called antrum. Right? Secondary follicle now gets transformed into a tertiary follicle. And what is the difference between secondary and the tertiary follicle? Now tertiary follicle has got a cavity called antrum. Right? The theca of secondary follicle also gets differentiated into two layers, theca interna towards inner side and theca externa towards outer side. So, let's see in terms of diagram. This is primary oocyte. Primary oocyte, when gets surrounded by a layer of cells, gets transformed into primary follicle. Primary follicle, when gets surrounded by some more layer of cells and when it has its own covering, it is called theca. The structure is secondary follicle. A secondary follicle, when gets surrounded by some more layer of cells, and there develops a large cavity called antrum, and the theca is organized into two layers, theca interna and theca externa, and then the structure is called as tertiary follicle, right? Lekin inside tertiary follicle, you can see what is this? This is primary oocyte. Still, at prophase 1 of meiosis 1, this point should be very, very clear, right? At this stage, the primary oocyte within the tertiary follicle grows and completes its first meiotic division and results into the formation of a large haploid secondary oocyte and a tiny cell called as the polar body, right? So at this tertiary follicle stage, the primary oocyte is present inside the tertiary follicle completes its first meiosis, right? And first meiosis results into the formation of two cells, and first meiosis is always reductional. So the resulted structures are unequal. One is a large cell called as secondary oocyte, and another one is a very small polar body. And where is this division taking place? In the primary oocyte, right? Present inside the tertiary follicle. Okay, now at the same time, a tertiary follicle becomes transformed into a mature follicle or a Graafian follicle. So let me show you the structure of a mature follicle, right? This is the structure of a mature follicle. This is the secondary oocyte, the product of meiosis 1. Polar body we are not considering. Why? Because polar body is of no use, so very soon it will degenerate, right? This secondary oocyte, as you can see, has a layer, layers of granulosa cells. Then it has a fluid filled cavity called as antrum, which is filled with a fluid called as Leaker folliculi, and it has two layers, outer one theca externa, inner one theca interna. Okay, and such a follicle with secondary oocyte produced after meiosis 1 is called as a mature follicle or a Graafian follicle. Okay, so what is the difference between all these follicles? First of all, once again we are coming to that point only because this is the most confusing part. The single cell structure is primary oocyte and it has developed from oogonia. Those oogonia which have entered meiosis 1 but has stopped at prophase 1. They are called as primary oocyte. Primary oocyte, when gets surrounded by a layer of follicular cells, it is called as primary follicle. When primary follicle gets surrounded by some more layer of granulosa cells and it develops its own theca or covering, it is called as secondary follicle. And when a secondary follicle grows and develops a cavity, antrum inside it, and two coverings or two theca interna and theca externa, it is called as tertiary follicle, right? And when a primary oocyte inside a tertiary follicle completes meiosis 1 and gets divided into two unequal structures, a large secondary oocyte and a polar body, then such a follicle which contains a secondary oocyte is called as a mature follicle or is called as a Graafian follicle in honor of the scientist who studied this structure. Okay, now this Graafian follicle is ready to undergo ovulation. What is ovulation? The rupture of mature follicle, the rupture of Graafian follicle, and release of egg from the ovary. At which stage? At secondary oocyte stage. And once again, I want to remind you people, secondary oocyte stage means only meiosis 1 has completed, right? Secondary oocyte enters now meiosis 2. Got it? It passes through prophase 2, enters into metaphase 2, but does not divide further. Why it does not divide further? Because it does not have its own centriole to form the spindle apparatus. So this has to wait for the entry of sperm, right? The proximal centriole of the sperm, we had read in this video only that the proximal centriole of the sperm completes second meiotic division of the zygote. So we are talking about this second meiotic division of the zygote, means we are talking about secondary oocyte. The egg has been released at secondary oocyte, so the egg waits for the entry of sperm. Okay, when the sperm fuses with the egg, then only it metaphase continues, metaphase 2 is completed and anaphase 2 starts, so that secondary oocyte divides completely into two unequal cells again, one large ooted and a second polar body. In humans and most vertebrates, the first polar body does not undergo meiosis 2. Likewise, the second polar body produced after meiosis 2, that too does not undergo. So we can say that polar body is of no use, right? These polar body degenerate at the end of the division, right? So let us now have a look of a diagrammatic representation of oogenesis. This is oogonium or the egg mother cell. Ploidy is diploid. This oogonium grows and gets transformed into primary oocyte, right? Once again, what is primary oocyte? A structure which has entered meiosis 1 but has stopped at prophase 1 only, right? And this primary oocyte is present inside the primary follicle. Primary follicle carrying primary oocyte gets transformed into secondary follicle. Secondary follicle carrying primary oocyte gets transformed into tertiary follicle, and this change starts with the onset of puberty, right? With the onset of puberty, the primary oocyte present inside the tertiary follicle completes meiosis 1 and forms two unequal cells, a large secondary oocyte and a very small polar body, right? This secondary oocyte only is the egg released from the ovary, and the process of release of egg from the rupture of the mature ovary is called as ovulation, right? Secondary oocyte then undergoes meiosis 2, though we have studied that it does not complete meiosis 2 immediately. Rather, meiosis 2 is also stopped at metaphase 2, and there it waits for the entry of sperm, right? That means if the sperm will not come, then the egg will get destroyed at meiosis at metaphase 2 only, at secondary oocyte stage only. There won't be the formation of ooted, right? But if sperm comes, then it will complete meiosis 2, and that will help in the formation of two more haploid structures, a large ootet or the ovum and a small polar body, right? And all these polar body, they are of no use, so they will degenerate, okay? And only this ovum the nucleus of the mature ovum will fuse with the nucleus of the sperm to result into the formation of a zygote. Okay, so this is end for today's video. Thank you children. If you have any problem, you can call me.